Well, I was hoping to be able to give this lesson to you directly, but with another no bus day here, I'm in the school today and making some more video lessons. And so this, I guess, will be your first lesson. I thought I'd be there for this lesson, but the idea of these video highlights or summaries is so when you go through the slideshow together as a class and Mr. Allen will walk you through those, I want to highlight for you some of the really important things and just make sure the things that are emphasized come across and you'll be all ready for our test. So earlier we went through and looked at protein function, and we looked at these four protein functions, and proteins again are king, and that is one of the big ideas for our course. But as we go through proteins, one of the critical things that they do is direct and control chemical reactions. And we're going to focus on this today, this idea of directing and controlling chemical reactions, as that is the role that enzymes play. And we want to look specifically at enzymes and how they work and how they're regulated. Well, enzymes are absolutely vital to our body, and the individuality of any given cell in your body is due in large part to the enzymes and the unique set of enzymes that that cell is producing. These enzymes do everything. They do control all the chemical reactions, and whether it be from building muscle to producing different enzymes to break down food to bolstering our immune system to processing energy for our body, enzymes do everything and control these reactions. Without these enzymes, the reactions in our body just wouldn't happen fast enough to support life. Beyond our body, enzymes are also crucial, and we take advantage of them in the food industry through baking, winemaking, all kinds of different things in the food industry. We maximize our use of enzymes. We even use them in the cleaning industry, and if you check your cleaning chemicals at home, I bet you find some of those cleaning chemicals have active enzymes in them that we take advantage of. So we want to look at enzymes more today, their role, how we regulate them, and also keep that idea that chemistry drives the biology. And so the unique structure of these enzymes is absolutely critical to their specific function. Because if one of these enzymes is absent, it's defective, results in a disease that could be mildly annoying, like lactose, to fatal. And there's a number of diseases where enzymes are not present and it results in death. So as we look at the specific characteristics of enzymes, understand that all enzymes are proteins. Not all proteins are enzymes. Proteins can do a lot of different things, but all enzymes are proteins. And these enzymes have three specific characteristics that we need to keep track of. These enzymes increase the rate of reaction. They act specifically on one reactant. So they are very unique to a specific reaction. And then these enzymes can be used over and over again. An analogy that's often used is that of a key to get into your house or something like that. It increases your rate of reaction. That key will only open that one door, and you can use that key over and over again. From a chemistry perspective, every reaction needs a certain amount of energy to get things to happen. And so if you see on the left-hand side of this diagram, there's a certain amount of energy that the reactants would have. But in order for them to react and come together, there's an activation energy that's required. And if you've taken chemistry, you've probably looked at that a little bit. You can see here the difference between that red line and that black line is that nearly half the energy is required when we use an enzyme. Ultimately, energy is required, but much less energy is needed for when enzymes are present, and that's what allows these reactions to go faster. So a model that's often given to describe enzymes is the lock and key model. You can see in the top of this diagram, the lock and key model shows how the substrate, what's being reacted, enters that enzyme, and that's how a reaction would take place, and either those products are going to be broken apart, or we might have a couple things coming together and we're trying to build something bigger. Enzymes can break things down, maybe as in this diagram, but they can also build things up. So a lock and key model has been around for a long time and would be what I was taught, I remember when I was a student. What's becoming more popular now in our understanding is what's called the induced fit model. And the induced fit model is more of a hand in a glove. It shows that there is a dynamic change conformationally in the shape of the substrate and the enzyme to maximize the binding that they have, and that optimizes the efficiency. So it's not as rigid as a lock and key model was traditionally. It's a bit more dynamic in its process, and so that lock and key model is still valid, but it doesn't quite encompass kind of the conformational changes that would take shape for both the shape of the substrate and the shape of the enzyme itself. Hopefully you can appreciate how important these enzymes are. Each one of these enzymes is specific to a reaction that's required. required by your body to survive. It has to happen in a timely manner. We have to have enough energy, resources, and time to be able to effectively respond to our environment. And so when we look at this, not only is enzymes important and absolutely crucial for life, and the unique set of enzymes is crucial to individual cells, but we also have to equally regulate these enzymes. We can't just have reactions occurring whenever we want, however we want. Life wouldn't respond that way. Life is ordered and structured, and we balance at the cellular level and as whole organisms the time, energy, and resources that we're given. And as we manage that time, energy, and resources, that allows us to critically respond to our environment. And if you can't respond to your environment, you can't survive for very long. And so enzymes are critical for those things. We're going to look at the next few is how we actually regulate enzymes and managing this time, energy, and resources to allow us to respond to our environment. So make sure that you get not only the importance of enzymes, the uniqueness of each enzyme to the reaction, but how important it is to regulate them. So enzyme regulation functions through basically 4 different levels. Through the environment, we can control enzymes. Don't forget that enzymes are a protein. And so like any protein, enzymes can be denatured. We would have done a lab if we'd had more time where we look at the effect of temperature on enzymatic reactions and to find an ideal or optimal reaction time. We could also change the pH to see what the optimal reaction time would be. So because they're proteins, we can regulate things through the environment by denaturing proteins. And so there's an ideal window where we can have enzymes function. We can also regulate enzymes by even how much of the enzyme do we produce. Do we have a lot of the enzyme or a little of the enzyme? How much of that reaction do we actually want? And so we can regulate them that way. We can regulate them on how much of the substrate we expose it to, right? How much reaction is available for breakdown. In chemistry, we talk about this idea of a limiting reagent. And it's the same idea with enzymes. We can control the amount of the enzyme, the amount of the substrate to help regulate that through the environment. Secondly, we can regulate things through cofactors or coenzymes. That might be like vitamins, minerals that we take in our diet. And these function on the enzyme to almost act like a saddle on a horse to just help that reaction site. And these cofactors, coenzymes, they help activate, if you will, those enzymes so the reaction can take place. We'll look at enzyme inhibitors that will actively lower the rate of reaction. And there's 2 types of inhibitors that we can use that would slow down the rate of reaction. There's competitive inhibitors and there's non-competitive inhibitors. Competitive inhibitors would be like, and I tell a story in the class normally about a girl that I liked, Leslie, in my first year university. The competitive inhibitor was the fact that she had a boyfriend. And while I waited and thought that eventually they would break up, they never broke up, and I think they're happily married today as far as I know. But there was a competitive inhibitor that blocked a potential reaction. And so competitive inhibitors, they bind to that active site and prevent a reaction from occurring. Non-competitive is a little bit different, and I normally would tell a story about a girl that I liked, Julie. Again, heartbreaking story as it is, but by the time I got the courage to kind of pursue a relationship with Julie and built up my nerve, I actually found out that she was with another guy, and what I didn't know is that she had her friends that were actively trying to set her up with another guy. And so that was a non-competitive. I didn't know about this other guy. We weren't in direct competition, but her friends were working behind the scenes to change that active site and prevent a reaction from occurring. And so you can have competitive and non-competitive. The stories probably make more sense when I'm there, but that's how I normally remember it, and my class normally remembers those girls' names, Leslie and Julie. So those are the 3 ways. The 4th way would be allosteric regulation. And allosteric regulation notices that enzymes can have active and inactive forms. In grade 11, you saw that pepsin and pepsinogen, there was active and inactive forms. Trypsin and trypsinogen had active and inactive forms. So enzymes as well can have active and inactive forms. And we don't want an enzyme to just go back and forth whenever it feels like, whether it's active or inactive. We want to control when it's active, and so we can have allosteric activators. And we want to control when it's inactive, and we can have allosteric inhibitors. These kind of work similar to the non-competitive inhibitors. The idea is that we would hold with an allosteric activator, hold it in its active position, much like a door wedge would hold that door open so people can come in and out of the room. An allosteric inhibitor would almost be like if we put a lock on the door. So when the door is closed, we're inhibiting that reaction and we actually lock it in place. And so those 4 ways are how we regulate enzymes in our body. And again, that's crucial as we respond to our environment. We have limited time, energy, and resources, and enzyme regulation is critical for life. I hope this video summary is helpful as you go through the lesson and make sure that the key things are really taken in. Make sure, again, you see things through the lens of how chemistry drives the biology, the unique chemistry of these proteins that act as enzymes, and the structure of them is critical. If we denature an enzyme, it no longer works. It doesn't regain its function again. The chemical structure is critical to the biology, and so keep that big idea in mind and understand the uniqueness of it. You also need to be able to make sure you can explain to me when we get to a test how we regulate enzymes and the different ways we regulate enzymes, and so make sure you take notes on each one of those. Good luck.